This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 456, recorded on August 25th, 2017. This episode is brought to you by the Defense Threat Reduction Agency. Part of the U.S. Department of Defense, the agency's Chemical and Biological Technologies Department hosts the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Find out more at cbdstconference.com. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello, Vincent. How are you?
Well, I got 25 Celsius, mostly cloudy. And what I've noticed is the nights are starting to get cool.
Ooh.
But it can't be. It's too early.
Yeah,
Something's weird.
No, you'll get it back. I have here 85 Fahrenheit, that's 29 centigrade, and entirely cloudy with a high probability of hurricanes.
Wow. Hurricanes. Yeah, something's brewing out there, I heard.
You bet. Harvey is going to make landfall around Corpus Christi, Texas, tonight at about between midnight and 1 o'clock as a Category 3 hurricane. And so that's not good. That's going to cause a lot of damage.
And that's going to stay in your part of the woods. It's not going to come up the East Coast probably, right?
Well, who knows? You know, all the predictions, obviously when it makes landfall, it then degrades into a tropical storm, and most of it has it hanging around down in the south. But who knows? I don't have much experience with hurricanes coming at me from this direction. I thought I was done with this.
Nope.
Okay. They seem to follow me around.
Do they ever get serious storms in Austin?
Oh, yeah.
They do. Huh?
They're predicting for this. We're 250 miles inland, but they're predicting 35-mile-an-hour winds and 7 to 10 inches of rain. So, you know, that's not child's play, but we'll be fine. You don't
have any trees or anything, right?
No. That's, you know, well, actually, we do live in the hill country, so there's a lot of trees around, but not like Florida. You remember Gainesville, we had these giant trees surrounding the house, so something like this would come through and you'd just be shaking in your boots because so always the threat that one of these is going to come down on the house.
Well,
All right.
Good luck. Good luck.
Thank you. It'll be fine.
Also joining us today from Madison, New Jersey, Brianne Barker.
Hi, everybody. In Madison, it is 74 degrees, pretty close to in the city. So that's 23 Celsius.
And welcome back.
Thank you. It's great to be here.
You were here. I don't know. When was it roughly? You must remember, right? You were here.
It was when I was on spring break. So March.
This year. Okay.
Yes.
Holy cow. Was I on board then, Brianne?
Maybe you weren't here that day. I'm not sure.
Because, you know, I am not trusting my brain here. It's got big holes in it. There we go.
So it was 4:48. And let's see if I can get a list of the participants here. TWiV 440. No, it can't be 448. That was Mavis. What am I talking about? I don't know. Oh, it's just those Twitter stuff. Let's try it again. I searched and didn't show up. Oh, here we go. Guests. I should know. I keep a page of guests on TWiV. And here we go. You're way at the top because it's 432. And was Rich Condit there? He was not.
Whew.
So that's why you don't remember.
That's a relief. Okay.
Well, nice to talk with you.
Nice to talk with you, Brianne.
And we saw you, Brianne, at ASM Microbe, which was in New Orleans as well, right?
Yes, I had 3 students with me at ASM, and I was also at ASV.
Now, you at ASM, I remember you got some very cool black lab coats.
We did. ASM was giving out some black lab coats to market one of their apps, and I managed to get them for myself and all 5 students in my lab.
Are any of them using them in the lab?
We all use them.
That must be cool.
It is.
That's something different. I like those. Those are very nice. Yeah, I know.
You can always pick us out.
It was the end of the day. They were trying to get rid of them.
Yep.
Well, cool. Welcome back, and you're going to be here next week as well, I think.
I will. Yes.
Good, and you can join us as frequently as you want. Is your school back in session there?
We start on Monday.
Wow.
I'm teaching a first-year seminar, and I met my first years this morning. But we start actual classes on Monday.
Well, they're going to really think you're cool because you're on TWiV.
Well, especially since I'm teaching virology this fall.
Very good. You have to give them assignments or something. Make them listen,
I will.
Especially to your episodes, right?
Absolutely.
All right.
They don't hear enough of me.
Of course. So let's see. We're bringing we have 2 kids left in college. One is now working. The 2, we're bringing them up this weekend upstate, and they're starting Monday.
Oh, wow.
Yep. Goes quickly. Boy, summer went by really fast. We have a couple of follow-ups. First one's from Jenny, who writes, "Hi, TWiVers. I have a follow-up for episode 455 in which you mentioned the film Unrest. The film is not yet available, but it opens in select U.S. cities on September 22nd, including in New York at the IFC Center. The film will also air on PBS Independent Lens in January 2018. Kathy wondered how Jen Brea was able to make the film, given that she has ME. Jen used technology to her advantage, sending film crews out to film interviews which she herself conducted via Skype from her bed. I believe that a great deal of editing and other work was done online as well." Unrest has won prizes at Sundance, River Run, Nashville Film Festival, and other film festivals. I had a chance to see the film and will be writing a review soon. I hope you'll be able to see it in theaters or on PBS. Cheers, Jenny. Well, that's how you do it. If you're bedridden, conduct interviews.
Plan.
You bet. Probably takes a lot of people. Well, most films do anyway, right?
Yeah.
Yeah, it's a big enterprise. The credits now just always boggle my mind how many people are involved.
Oh, it's huge. Totally huge. In fact, too many people. I was looking at some critique, and they were saying in the old days there were not that many people involved in films, and now a lot of those people are like special effects and animations and all that sort of thing where they have just thousands of people. Just amazing. Well, evolution, it happens. Rich, can you take Anonymous?
Anonymous writes, "Thank you for reading my message on TWiV 455. Nothing particularly interesting has happened in the Voinette saga. Is that how I pronounce his name? Voinette saga. Since you discussed it 2 years ago on 347, TWiV 347. The Scientist published a piece on the new paper/editorial. That's what we discussed last time. And summarized some of the history and gives a link to that. Many papers have been retracted since the ETH Zurich investigation concluded, especially at the EMBO Journal. There were 5 papers retracted. Most papers, he says 22, were corrected rather than retracted. Voinette has kept his position at ETH Zurich, and new papers from his lab have started to appear. PubMed search indicates that he has co-authored at least 5 papers in the last year. I'm not sure whether Voinette deserves his second chance, but many of his trainees presumably did no wrong, so I'm glad that they are successfully publishing at least some of their previous work. However, Patrice Dunoyer, or how would you pronounce that?
Dunoyer.
Patrice Dunoyer, the main subject of the Nature Plants editorial, was not an innocent bystander. He was first author on many of the retracted papers, including the single most inexplicable case of band and lane duplication, and was therefore responsible for either misconduct or extreme sloppiness. This fact added some controversy to the new editorial.
There's a PS.
Yeah. Leonard Snyder has written about the case extensively in his blog, but I and other plant scientists do not like his tone, so I would not recommend linking to it. We can leave that to the listeners to find out.
Now they get his name. I think this. So I was reading some of this blog by Schneider, and the article that is the most recent is entitled "Voinet's sidekick Dunoyer welcomed at Nature Plants despite retractions and admitted misconduct." The Olivier Voinet scandal of almost 2 decades-long research misconduct and data manipulations has reached its logical conclusion. The French plant pathogen researcher and everyone who helped him manipulating and publishing dishonest and occasionally retracted papers were either forgiven or declared as fully reformed. The siRNA co-discoverer Voinet, who cynically put was too big to fail, remained professor at ETH Zurich and kept his ERC funding. He is meanwhile back to publishing in exactly the same elite journals where he had to retract and correct papers for manipulations. Of all his partners in crime, only his dependent right-hand man or sidekick, Patrice Dunoyer, was ever investigated, and as punishment was suspended for an entire month by his French employer CNRS. His lab was about to be dissolved, but the Nature Publishing Group came to rescue and accepted his paper just in the nick of time. The accompanying editorial in Nature Plants, which we read last week, written by the chief editor Chris Surridge, can only be described as bizarre and is titled "Give Research a Sporting Chance." Surridge, who apparently sees data manipulation as a professional sport in a race with doping detectives. Oh boy.
I can see why they don't like his tone, but I don't like the whole thing.
I don't know why you let everybody back without any kind of...
No, not at all.
Just don't let them back at all. Forget it.
I mean, once people do this, it seems to me that it's going to happen again.
Everybody thought, some people thought that the presidency would change Donald Trump.
No.
You know, the leopard seldom, if ever, changes its spots.
No, not at all. So, well, I just would worry about anything coming out of him or some of his people. We'll see. We'll see if there are any issues, but it seems a bit unfair, right?
I don't like it.
Especially all the people that are good that don't do this out there, and it's hard enough for them. And yeah. So thank you for that update, Anonymous.
Mm-hmm.
All right, we have a snippet in a paper today, and it turns out, this is just totally serendipitous, that they're related.
They are, and they're both related a bit to what I do in my lab.
Well, that's even better. Now, I know the paper that we're doing, you had asked about a couple of weeks ago, and I said, we'll do it when you come on.
Mm-hmm.
But the snippet was something I was looking at for last week's paper, and last week we did a paper on mutations in young kids with severe respiratory infections, and they identified mutations in the gene encoding MDA5. And I had found today's snippet paper at the same time, and I said, we'll do it as a snippet next time, and it'll be an ARC. But little did I realize that both papers would be part of the ARC today. So the snippet is from the Journal of Clinical Investigation, and I just found this totally fascinating. Inborn errors in RNA polymerase III underlie severe varicella zoster virus infections. This comes from many, many authors at many, many institutes in throughout Europe and even here in New York City. And the first author is Benson Ogunjimi, and the last author is Trine Mogensen. And what
most of the work is done in Belgium.
Belgium.
At the University of Antwerp, from what I can tell.
Okay,
so as you say, many authors all over the place.
Huge. And no yes, the first two authors. Let's see. Soren Paludan and Trine Mogensen contributed equally. So those are the two senior authors.
Yes, and Soren Paludan is in Denmark.
Okay.
doing a lot of work on DNA sensing.
Also on this paper, the third to the last author is Jean-Laurent Casanova, who's here in New York City at Rockefeller and is well known for doing this kind of work as well. And he, I believe, handled the PNAS paper last time, if I'm not mistaken, if my memory serves me well.
And the note also says that the first three authors,
contributed equally.
Ogunjimi, Zhang, and Sorensen,
right,
contributed equally.
I just didn't even see that. What am I doing? All right, varicella.
Actually, on the, I get frustrated because on the online version, it's really hard to find where those footnotes, where they're from, and et cetera.
Yeah.
On the PDF, it's right there.
Yep. Yeah, probably online. It's all the way at the end, right?
I couldn't even find it.
All right, so this is a snippet, so we'll get through it relatively quickly. Varicella zoster virus infects you when you're young, very young. You get chickenpox, primary infection. And then the virus becomes latent, the genome becomes latent in peripheral ganglia. And then when you get older, typically, but it can happen sooner, when you get older, 50 and above, you get reactivation, which results in zoster or shingles, which I have had. Have you had it, Rich?
I have not had shingles. No, thank you.
Interesting. And you're older than I am.
Hey, not every, I got good genes. I got really long telomeres. Okay. I really won that lottery. I have tried to poison myself all through my youth and I was unsuccessful. So I'm going to start taking care of this thing. So I think, God, you had shingles.
Yeah, I had shingles way back at the beginning of TWiV. And I think around episode 8, we had Saul Silverstein on, the herpes guy, who's my colleague here, and we talked about it. And yeah, it was interesting. Shingles on my chest along one dermatome in one area, innervated by a single nerve, and just a rash, And I didn't do anything. I didn't see anyone or take anything. It lasted about 2 months, and it was just kind of annoying. It wasn't terribly painful or anything. I
think some cases of shingles are really bad. And
yes,
really
yes,
painful.
For sure.
And then, of course, a big problem is post-herpetic neuralgia.
Yes.
Where the shingles resolve, but there's some sort of nerve damage or something that lasts. And so you don't have any agent that you can take care of, but you've got pain that can persist for a very long time, if not indefinitely.
This is, of course, a herpes virus, these large double-stranded DNA-containing viruses. And this paper deals with the primary VZV infection, which in some cases results in a more severe disease than chickenpox, like central nervous system disease, pneumonia, even stroke. All right? And the frequency is kind of low, 0.02 and 0.025% of CNS and pneumonia in kids. What were you going to say, Rich?
I just wanted to make a note because this was important to me as I was reading this, make a note on the pathogenesis of this virus, how it gets around. Because you acquire it as a respiratory infection.
Right.
But obviously, ultimately, it makes it to your skin. So in the meantime, the way it does that is to travel through your blood. And the way it travels through your blood is to infect immune cells, specifically T cells. And travel around through your blood and then ultimately make it through your skin. So it's infecting epithelial cells in your respiratory tract and then T cells and then I think both endothelial cells and maybe epithelial cells in your skin. I'm not sure. But importantly, a major target are lymphocytes. That becomes important for this paper.
So the question here is, why do these kids get more severe varicella? And there's no indication that the viruses are different. So just like last week, we look at the host, and they did whole exome sequencing. They have 21 children with severe acute varicella zoster infection. And eventually what they found, and this really opened my eyes, mutations in RNA polymerase 3.
Yeah, this took me by surprise, but of course, if I had been awake, I was just, if you look up the references.
Yeah, it's all there.
Pol3 as a sensor has been around since 2009.
Yep. And Brianne, you must have known this, right?
I did, but Pol3 as a sensor seems to be very specific to AT-rich DNA.
Yeah.
And so It ends up signaling really through the RNA sensing pathways.
Right.
Sure.
So it's an RNA polymerase, but it recognizes DNA templates, right?
Exactly. So in my lab, we have to be very careful about the DNAs that we use as controls to make sure that we are always hitting the DNA sensing pathways and not the Pol3 RNA sensing pathways.
So this is a cytosolic sensing or nuclear?
Cytosolic.
And do we...
wait a minute. Let's do RNA polymerase 3 here for a second.
Yeah.
Because so there are... Cells have 3 different RNA polymerases. RNA polymerase 1, which transcribes ribosomal RNAs. RNA polymerase 2, which is responsible for messenger RNAs, is doing really the heavy lifting. And RNA polymerase 3, which does tRNAs and 5S RNA that's in the ribosome. And what else?
The small stuff at the bottom of the gel.
MicroRNAs. Is it 2 microRNAs?
I don't know.
Some, at least. But
small RNAs.
So it's always been sort of the, poor cousin to the big guy, Pol 2, right?
Now,
and I would think it would be in the nucleus.
Yeah.
What's it doing in the cytoplasm?
That's exactly it.
So, that is a important general question for a lot of DNA sensors. The location of the sensors and the location of the DNA is frequently a little confusing in terms of whether it's nuclear or cytoplasmic. A lot of that's not really clear, but RNA Pol3 can very clearly sense DNA that's transfected into cells, which we can show is in the cytoplasm.
Is this something you work on in your lab, Brianne?
I work more on the CGAS and IFI16 sensors that are mentioned here and in the other paper, but we do think a lot about sort of avoiding the Pol3 sensors with our controls.
So IFI16, is that a nuclear sensor?
It is a nuclear sensor. Where
CGAS is cytoplasmic, right?
Yes, that is the general idea.
Now, you mentioned that the Pol3, does Pol3 mainly transcribe AT-rich DNA, or does it just, do all the templates for Pol3 are not just AT-rich, are they?
They aren't. It was first sort of shown to be involved in sensing of a molecule called poly-DADT that a lot of people were using for their DNA sensing experiments. And so people were using poly-DADT for DNA sensing experiments and suddenly realized that cells with knockouts in the RNA sensing pathway seemed to have a poly-DADT phenotype. And that was how this whole RNA Pol3 link was shown. So
if you knock out RNA sensing, you didn't respond to poly-DADT.
Exactly.
Right.
Which you wouldn't expect if it was a straight old DNA sensor.
Yeah. Right. The literature, as we'll probably get into more later, really thinks very separately about RNA sensing and DNA sensing.
Yeah.
And we're seeing a few places where they interact.
With respect to this cytoplasmic localization, in this particular case, it doesn't really matter because VZV, I would expect it to be nuclear in this case because VZV replicates in the nucleus. So Pol3 can be in the nucleus and see VZV DNA and everything's fine, except, of course, I guess, that the rest of the sensing that we're talking about is cytoplasmic.
Exactly.
So
whether sensors translocate and what exactly induces the translocation is part of that information that's up in the air.
Okay, so when we get this whole pathway together from this snippet, we can maybe come back to that.
We can try.
I think it's not clear. I've always had issues with DNA sensing in the cytosol because most DNA viruses, they put their genome in the nucleus. So I think there's still a lot more to learn. IFI, what is it, IFIH1?
IFI16.
IFI16 makes sense because it's in the nucleus, and that's
exactly
great, but I don't understand CGAS in the cytosol, although the second paper might start to address that, at least for RNA viruses.
Yeah. My short answer to that is that there are a few recent papers that have talked about how IFI16 and CGAS can collaborate.
Yeah, right. When I spoke with Ileana Christea at Princeton,
a long
time ago, she said, there's evidence that they're collaborating. That was a couple of years ago already. Yeah. All right. So in this, they find in a couple of these kids 3 heterozygous missense mutation in one subunit of Pol3A, and 3 kids, and then 2 heterozygous missense in Pol3C, another subunit, and then 2. Sorry, I just didn't get that right. But there are changes in 2 subunits, and they're heterozygous and they're missense, which means they stop translation. And one kid, I think, is heterozygous for a change in both subunits.
No, wait a minute. You said missense. I mean, you get a new amino acid.
New amino acid.
Translation.
That's nonsense. Oh, that's a stop gain. That's what they said last week.
Yeah.
They said it in this paper too.
Stop gain?
Yep.
Too bad.
I think that is becoming part of the vocabulary.
Yeah, we can't.
My guess is that they call these mutations stop gain in the computer programs and stuff that are used to analyze this. I'm just guessing, but when I see it in a couple of totally independent sources, it's obvious that that's what's happening.
Right.
So we got 2 different subunits involved here.
Right.
Distributed amongst these and this enzyme has like 17 subunits.
Yeah, that's crazy.
So we got Pol3C and Pol3A.
Right.
And just to recap, everybody is heterozygous no matter what they got.
Right.
2 of the patients have just 1 mutation, and 2 of the others have 2. In each case, 1 in Pol3A and 1 in Pol3C. And as I look over this list, so we're talking about a total of 6 different alleles, if you like, 6 different mutations, and no 2 of them are the same.
Right. And 4 out of the 21 patients have changes of some sort.
Right. And the way they did this was to, do exome sequencing, and then they kind of they filtered that by looking amongst those for mutations that exceeded a rare allele frequency, and among a list of enzymes that might be involved in immune sensing, including Pol3.
Right.
So they had a candidate gene list. Okay. So we don't know what's going on. This is 4 out of 120 kids. So, and I don't know that they even looked for splice site mutations, did they? Yes, they did. So we don't know what's going on with the other 120 some odd kids, but it could be anything.
Lots of other things too. Yeah.
Mm-hmm.
Yep.
All right.
I think it's important that they did double check to make sure that none of the other known DNA sensors were mutated in these kids.
Yes. Right.
Yeah. I think I want to skip ahead to, there's a lot of interesting clinical information here, but I don't want to go into it unless either of you do. I wanted to talk about the nice experiments where they took PBMCs from these kids or from healthy controls and show that there's a problem in them. And that's the experiment I wanted last week. And,
so PBMCs are peripheral blood monocytes. Okay. So that's white cells. Okay. Which is why I thought going over the pathogenesis was important. These are cells that are normally targeted by the virus. And
here, Brianne, they're using poly-DADT as an inducer, right?
Yes, exactly. They're using both poly-DADT and poly-IC, which is a classic RNA stimulus to hit the RNA sensors.
And so these are synthetic double-stranded DNAs that they're sticking into the cells to see what happens.
DADT is a synthetic DNA. Poly-IC is a synthetic RNA.
Right.
So this shouldn't change, and changes in Pol3 shouldn't respond to changes in poly-IC, right?
Exactly.
Yeah. So they use a Pol3 inhibitor, all right? Something obviously that's been around that's known to inhibit the enzyme. And they're using interferon expression as a readout of poly-DADT stimulation. And the inhibitor can block interferon expression induced by poly-DADT. So that's pretty cool. This is from wild-type PBMCs, right?
Mm-hmm.
And the bottom line, I think, and you can give more details if you want, is that the PBMCs from the patients are deficient at responding to poly-DADT, but not poly-IC, right?
Right, exactly.
And again, looking at interferon A, B, and et cetera. So these changes are impacting the response to TADT. What do you think of those experiments, Brianne? Are those good?
I think they look like great experiments. I'm really happy with them, and I think it is pretty clear what's going on with the patients. In a couple places, they also use htDNA, which is herring testes DNA, and that more traditionally stimulates the other DNA sensors. And they don't really see the same dramatic response with the htDNA.
Now, the next thing is kind of relevant to something we talked about, and I had forgotten this. Pol3 initiates this transcription from DNA regions with high AT content. Okay, so I guess all transcription by Pol3 is preferentially initiating at high AT. That's interesting. Which would ask me then why. I would then ask the question, why don't we respond? Why don't we turn on this sensing pathway with our transcription, right?
And that is a great question.
Which we don't get an answer to, right? Yeah.
No.
But anyway, they found that the VZV genome has an AT content of 54%. And they're islands of high AT content. And in contrast, herpes simplex virus 1 and 2, Epstein-Barr, have low AT. And I know that because my colleague Saul Silverstein always complained about the high GC content of herpes simplex DNA. And so they show basically that VZV, high AT DNA is good at stimulating interferon when you put it into cells, and the low AT DNA is not. These are with PBMCs from controls or patients. Mm-hmm. The patients are impaired at responding to the high AT, but not, the low AT doesn't do anything.
Right.
Okay, next, as Brianne said earlier, Pol3 takes this AT-rich DNA, it makes RNA, and it has a triphosphate at the 5' end.
Which is somewhat unusual. It's not capped. Okay,
right,
so that's the triphosphate left over from the first nucleotide. Excuse me, Brianne.
Oh, I was just going to say, so that's part of why we wouldn't respond to some of our transcripts. Because some of our transcripts are capped.
Right. And the ones that are with the triphosphate, there are some on our cells, but they're in the nucleus, right?
Mm-hmm, right.
And so we don't respond.
This may speak again to the nuclear cytoplasmic thing.
Yes.
Yeah.
Could be a compartmentalization thing.
But Brianne, this triphosphate 5' end is a substrate for RIG-I, right?
Yes, it is.
And so they transfect poly-DADT into cells, or they put VZV high AT DNA into cells, and they can measure interferon production. Actually, they extract RNA from these cells, and they put that into cells, and that stimulates interferon production.
Mm-hmm.
The idea being that the Pol3 is transcribing this DNA and making a RNA that hits the RIG-I pathway. And this happens with wild type. It's deficient in donor cells from donors with these mutations in them. Right, so they don't make the conclusion here is they don't make immunostimulatory RNA.
Right.
Presumably the Pol3 is defective, right?
Right.
Mm-hmm.
Which of course begs the question of, it must be good enough for their survival, right? So this is a selective change that doesn't impair Pol3's function in the cell, but it's impairing sensing.
Well, one thing they did was they also, elsewhere in the paper, I believe, measured how much Pol3 these patients have and how much of the normal cellular Pol3 transcripts they have, and all of that was normal.
Yeah.
So these are subtleties, and I see this transcription of sort of random AT-rich DNA as sort of, well, I don't know if you want to call it an artifact, because obviously this thing is working as a sensor, but it's a sort of an off-target activity of Pol3. Pol3, like most polymerases, has a very stringent and well-described initiation promoter, and it's got initiation complexes that help it do this and termination complexes. So the normal genes that Pol3 transcribes are well-defined and have very specific start and stop sites. This AT thing is sort of it's a sideshow, but an important sideshow.
Yeah.
Yes, absolutely.
Then they ask, is there any difference when you infect these PBMCs from either the patients or wild type with VZV? Now, you can't just add VZV to cells because you can't easily get much VZV, but what you can do is add VZV infected cells. And that's also something I heard for many years from my colleague who would always complain that you can't purify or get much. There's not much extracellular VZV, but they take MeWo cells, infect them with VSV, then they put those infected cells in with the PBMCs from the patients. And basically, there's reduced induction of interferon-stimulated genes from PBMCs from patients with the mutations in the Pol3 compared to wild type. So viral induction of interferon-stimulated genes, which is a result of, again, the RNA being made by Pol3 and stimulating RIGI, activating RIGI is lower in cells from the patient. So that's something you would predict.
Yes, although it certainly looks like there are some differences in some of the ISGs with different patients. Patient 2, for example, isn't as impaired.
Right. Right.
And they, at some point in this, we're coming to that. Sorry, I got it getting ahead of myself. Go ahead.
You're thinking about the other viruses?
I'm thinking about the fact that one of these two mutations in patient 2, I think it is, doesn't look like it's doing a whole lot. It's only one of them.
What happened to patient 2? Let me go back to the clinical, see if there's anything. So P2 is one of 2 Belgian boys, 3 and 5 years old, were seen in the hospital due to VZV encephalitis. P1 had encephalitis and P2 had cerebellitis. Also known as acute cerebellar ataxia, is a rare inflammatory process characterized by sudden onset of cerebellar dysfunction. Okay, so the cerebellum is the little thing at the back, right?
Yep.
So that's different from encephalitis is the main part of the brain.
Right. That's why I have neuroscience colleagues.
So P2 had cerebellitis and P1 had encephalitis. So I don't know if cerebellitis is less severe, but you're saying that P2 cells were a little bit different in their responses, right?
They were.
The thing I was thinking of is here, I don't know what part of the paper it is. I just finally picked it out. I don't know where the data are, but it says PBMCs from the mother of P4, who is heterozygous for the same R84Q Pol3 mutation as P4, but does not carry P4's other things. So mom has one of P4's 2 alleles.
Right.
Dad must have contributed the other one. Mom's PBMCs have a normal response to the VZV infection. So they're saying, well, maybe that mutation isn't doing much in P4.
Mm-hmm.
Okay?
Got it. By the way, we didn't go over this, but they have some parental genotyping. So,
well, I was very interested in that. Yes, they do have some parental genotyping, and there are SIBs as well.
Right.
And there are parents and SIBs who have the genotype that's the same as the affected children, and those individuals are not affected. Mm-hmm. And so they call it incomplete penetrance.
Right.
Okay, which is a sort of a general genetic term for saying that not everybody who has the mutation shows up with the phenotype. All right?
I really liked that they were talking about the parents' genotypes and phenotypes. I remember I was listening to your episode last week and sort of really wishing that was included.
Yeah. I also like these experiments where they take PBMCs and they characterize them functionally. They did not do that last week.
Mm-hmm.
Mm-hmm.
Now, maybe it's harder to get respiratory cells out, right? PBMCs, you just bleed. It's a lot easier. But I think if you can duplicate the phenotype in cells from the patient, it's better because that rules out that there aren't other genes that are involved that, might be contributing.
Mm-hmm.
This thing about incomplete penetrance really interests me because on the one hand, it could be, the circumstances under which you get this may be relevant. Okay. Like how old you are and, what, I don't know, other particulars of the infection. So it could be that these other carriers of this allele were never quite in the same circumstances, though presumably they've all had a VZV infection at one point. But it also suggests that this is subtle and other elements of a person's whole genetic makeup may impact on whether or not this particular phenotype is expressed. So it could be circumstances, it could be genetics.
Yeah, they mentioned that last week. It can be the dose of virus you get. Other genes might be involved. Other genes might complement, or
maybe other elements of your health status.
Yes, socioeconomic status, nutritional status, right?
Maybe co-infections.
Could we say the state of the moon? No, not the
Hey,
Did you see Did you see the eclipse, Rich?
I did.
You did?
I posted on the TWiV Facebook page. Did you not see that?
I didn't see it. No.
me and Neva were together.
Oh, how cool!
with Ib in Buda at the Buda eclipse party.
How cool!
It was awesome. We had a great time.
Did you have a totality?
No, it was like 50-60%, something like that.
Did you see it, Brianne?
I did. We all went to the observatory at Drew and used solar filters on our telescopes here.
Nice.
Cool.
So it was great.
Oh, now I see them. Yeah, Rich and Neva at the Buda eclipse.
And we did Neil deGrasse Tyson's colander thing. Okay, you can use if you have the sun shining through a colander onto a piece of paper, you get the colander pattern, but for each pinhole, you get the crescent shape of the eclipse.
Yeah, it's cool.
That was a big hit.
So you got Neva, who's in Austin, then you have Tom, who's also in Austin.
Yeah, and there's a couple other TWiV fans around here in Austin. So we got a real crew brewing here.
Well, that's good.
Yeah, it's great.
We can use that.
As a matter of fact, as long as we're going down a rat hole.
Sorry about
that. Remember my pick from last week was the Science Art Gallery.
Yeah.
So I communicated with the founder of that gallery and pointed out to her that we'd done this as a pick, and she thought that was cool and invited me to participate in some of their stuff. They do a monthly science happy hour out at the gallery, so I'm going to go participate in that. So this is all good. We're building a science citizen science thing here in Austin. It's great.
I was up at a Boston College retreat outside of Boston, and it was too cloudy. We couldn't see it. But what was cool was, and by the way, my hosts, Bridget and Welkin, shout out to them. They had it on the program. They had, talks, and then at 2:30, Solar Eclipse 2017. They had a half hour, and everybody went outside, and it was too cloudy to see anything, but everyone stood outside anyway. And then the 3:15 talk was me, and I figured nobody's going to come back in. But it was so bad that the room was full, and nobody wanted to stay outside. But I'm sure if the solar eclipse had been good, they would have stayed outside. I would have spoken to no one.
The next one in 7 years is going to be total in Austin.
I heard that.
So yes. I'm ready for that. Apparently, if you're in an area where it's total, it's really astonishing.
So Dixon drove somewhere in Tennessee, right? They picked up a small town on the map that was supposed to be in the zone of totality. They drove there, and he said there was no one there. They went into a field next to a high school. They lay on blankets, and they said they watched it get dark, and he said it was just amazing.
I'll bet.
And I've seen videos. I've seen videos of people from this eclipse in the field, and as it gets darker and darker, everyone's like, "Whoa! Oh my gosh!" You can hear them exclaiming and like freaking. It is. I've never seen one, but apparently it's amazing.
Yeah.
It's so cool.
It gets like seriously dark, and the corona shows up big time. So it must be amazing.
No, maybe I'll come to Austin.
So Austin is 70. Oh, wait, I wonder. Maybe that'll be. Let me see.
Oh, Rich, you know where another place that's going to have totality?
Where?
Buffalo.
Is that right?
I noticed that as well. My family is from upstate New York, and I noticed that upstate New York was going to have totality as well.
You know, Buffalo's closer than Austin.
in the 1 7 years from now?
Yeah, 2024.
Okay.
That's a weird pattern, I think, Austin.
That is a weird pattern.
Right.
But I think they are weird patterns. Well, I'll come back to that. There was a PBS show in the eclipse, a Nova. No, I won't come back to it. There was a Nova on the eclipse last week that was just awesome. It was really great. You know, I get sometimes disappointed by some of the science shows because they kind of water down the science and fill it up with a lot of gee whiz stuff. And that's not like what this was at all. This was hard science. And they followed several groups around looking at the eclipse. It was a great show.
This is why people love astronomy, right? It's this visual thing. It's happening in your real time. You know, we can make beautiful pictures of viruses, but they're static, you know.
So let me see. 7 years from now is another 350 episodes from TWiV. So we'll be at about TWiV 800. Maybe we can have the TWiV 800 party for a total eclipse.
We could do that, yeah.
Jeez, I'm going to count ahead, see if that works out.
Go ahead, go for it. You got it. You had 500, right?
Yeah.
All right, the next experiment they did, do other viruses have an issue with these altered Pol3? And as far as RNA viruses go, there's no problem responding to a panel of RNA viruses that they tested.
All the patients look normal.
Patients look normal. Herpes simplex virus, no. One patient, P1, had reduced interferon after infection with herpes simplex 1, but P2 and P3 were normal. So this seems, it really is VZV. And maybe other viruses with AT-rich genomes, who knows, right?
Right.
These cells, they co-culture them. Now, we've looked so far at co-culturing infected cells with these PBMCs and induction of ISGs. And then they say, is viral replication controlled? And basically, the PBMCs from these patients with the mutations in the Pol3 don't control viral replication as well as wild-type cells. And you can rescue that by putting a plasmid encoding the wild-type Pol3 into those cells. And that is, I think, a beautiful experiment.
I agree.
So by not controlling the infection as well, what we mean is that VZV grows better, apparently, or that their measure is gene expression from VZV.
Yeah, they're looking at a viral protein, right?
VZV apparently replicates better in the patient cells than in normal cells. So the patient cells are not tamping down the infection to the same extent as normal cells.
And the cool thing, when you put the wild-type gene on a plasmid and put it in, it fixes that problem. And
that's critical.
That's critical because that tells you that's the main problem, right?
Yep.
And that's what was lacking from last week's study. And because you can always imagine there's something else too. Anyway, so that's so for me, the amazing gee whiz thing here is not so much that they can identify changes in patients that get more severe disease, because we've seen that now for a number of viral infections, but that, as Rich said, we've been in the dark here. Pol3 is a sensor. I just didn't realize that because I stopped following the sensing field a long time ago, and I don't know, it never came up on the TWiV radar. I don't know why. So, Brianne, you said 2009 this is known?
2009, maybe a little after that, but around there.
I looked up there's one bit in the introduction here with a bunch of references. And it looks like key references go back to 2009, fingering RNase 3 as a sensor.
My excuse would be those are early days of TWiV, so we're just finding our place in the world. Now, one thing I thought of here is, and I don't know if they did this last week. Maybe you know this, Brianne. Brianne, if you then look in the database of all human sequences, do you find these changes in other people that haven't been studied because they have severe virus infections?
I don't know the answer, but I think that would be a really exciting thing to do, and I would love to know.
Because we got a lot of human genome sequences now, right? You can
exactly.
And I guess you could look at Pol3 and say, do we see SNP, single nucleotide polymorphisms, that may have similar phenotypes, right?
They also have a section in here because you can model the structure of Pol3. The yeast Pol3 enzyme, the structure is known. And so you can model these mutations onto that and see if you can figure out why they are having the effect that they have. And there's a whole lot of language in that paragraph that basically says, I don't know. Yeah. But what I like is in the end here, they say, since defects in Pol3 are associated with both degenerative processes in the CNS and susceptibility to VZV in humans, there is a requirement for more knowledge on the biochemistry of Pol3 in transcription of endogenous as well as exogenous types of DNA. And I can hear a bunch of Pol3 biochemists just cheering at that,
Yeah,
because this is a great example where basic biochemistry on, a regular old enzyme, I don't want to call it a loser enzyme, but it's kind of taken a backseat in some people's minds. It's not it's not like Pol2, okay? But the basic biochemistry of transcription of this thing is really important.
Serendipity,
and there's got to be
serendipity.
Yeah, absolutely. And another call out for basic research.
So, Rich, if you submitted a grant proposal today on Pol3, people would yawn unless you knew about the sensor part. Right? Because they say, what are we going to learn? And the thing is, who knows?
Right. So now they can all put this paragraph and reference this paper and put it all in their significance section of their grants. So I told you so.
So they say in the discussion, a couple of things I wanted to point out. So these changes in the Pol3, they impair transcription from AT-rich DNA, but not from normal non-AT-rich DNA. And no effect on 5S ribosomal RNA transcription.
Right.
So these are just random changes that come up, and if you happen to get infected with VZV, oh boy, you get a problem.
So, yeah, I think, like I said, I think that this particular activity of the enzyme, as I said, is kind of a sideshow. It'd be interesting to get somebody like Harmeet on this and find out whether, whether some of these sites undergo any sort of selection that would be significant for
that would be great.
Antiviral stuff, because clearly this mode of transcription for this enzyme is different than its day job, okay? Because the day job is not affected.
So, Brianne, they say in the discussion that C-gas is not a key sensor of AT-rich DNA.
Right. So it doesn't seem as though the AT-rich DNA is really going through C-gas, which is a little bit weird given that we typically think of C-gas and IFI16 as being sequence independent.
So if you take a cell where you've got these Pol3 changes and you put in AT-rich DNA, C-gas doesn't see it?
It does not seem to, no.
Oh, that's weird. So that's
RNA. That's Pol3 jobs. I don't have to deal with it. It's not my contract.
So that means that VZV at least is not sensed by C-gas, right?
Well, I don't know about that. I think that there may be some other parts of the VZV genome that may hit C-gas.
Okay.
So one of the things that occurred to me later on that I think is kind of amusing is that in adenovirus, there is a transcript called VARNA that is an immune modulator. It's a small viral RNA that interacts with, I guess, the PKR system to tamp that down and interfere with the cell's immune response. And that is transcribed by Pol3. I'd be interested to know whether Pol3 also senses VARNA, in which case it would be senses adenovirus RNA, or adenovirus DNA, in which case Pol3 would be doing two contradictory things on this particular viral DNA, making an immune modulator of cells and also sensing the presence of DNA.
Another interesting point here from the discussion. So they have a data set of 222 patients with herpes simplex encephalitis, and none of them have enrichment in these Pol3 mutations. So this is, again, HSV doesn't seem to be sensed by Pol3. They also suggest, and this is interesting, I think somebody mentioned this at the top of the show. So VZV requires a viremia phase, but not herpes. So they think that the susceptibility to severe VZV might be explained by the high AT content of the genome, but also low CGAS expression by blood cells. Brianne, what's that mean?
So most of these sensors are studied quite a bit in macrophages, and there's not as much known about them in other cell types. It does seem like CGAS expression levels vary pretty dramatically between cell types. And so I don't know CGAS levels myself in T cells, but it's pretty low in some other non-macrophage cells.
Got it. So that would predispose low CGAS expression by. So that means, so the possibility is that VZV might be sensed by CGAS and blood cells, but it's too low to do that.
Right. Exactly. So if those blood cells had more CGAS, then perhaps
right.
it would be being sensed.
Got it. Yeah, and a mutation in Pol3 might not have the same effect in young kids in terms of serious disease, right?
Exactly.
Cool. All right, one more thing. Their last sentence is clinical implications of these findings on, prophylactic VZV vaccination, acyclovir treatment, genetic counseling. So I guess if you're at the point where everyone's genome is sequenced, you have a new baby, they have these Pol3 mutations, I guess you could think about vaccinating, right, very quickly.
I guess.
As soon as the kid is old enough to take this vaccine. And if you did get chickenpox later in life, it really shouldn't, because you should get vaccinated.
Right.
If you happen to, you should be aware of the possibility of severe mutation to be ready with acyclovir, the antiviral that works, right?
Yeah, I think I'll just get the vaccine. Well, I've had chickenpox, but I would have just gotten the vaccine.
Yeah, I was too young. I'm too old to have gotten the vaccine. It wasn't around when I was a kid, so yeah, I did get chickenpox. Yep.
I am also too old.
Really?
But I got the shingles vaccine, which is basically just a double dose of the chickenpox vaccine.
Yeah, it's the old man vaccine, right?
Yeah, the old man, exactly.
When did you get that, Rich? That's why you didn't have shingles. You're like, I didn't have shingles. You got the bloody vaccine.
I don't know, 5-6 years ago, something like that.
Yeah, I was just at the age when I should have got it. And then I got it. I got shingles. But the VZV vaccine, I think we talked about this, and I think you were there, Rich, isn't great, and there's some new ones in the pipeline which are really awesome. Maybe it's a shingles vaccine that we're talking about. I only, if I remember this, I could be fabricating that memory. This is something, and part of that fabrication is, maybe part of that conversation was we ought to do a TWiV on this, or get a guest on. Oh, that's right.
Who knows about VZV and the vaccine and all? Because I think that the chickenpox shingles vaccine is a fascinating topic, because that's a vaccine that's going to establish latency. Okay, so that's something new.
So we talked about a clinical trial of a new vaccine, which is a subunit vaccine. Okay. Oh,
yes. And yes, I do remember.
Really, really good efficacy, like 100%.
That's right. Right.
And this is because the shingles vaccine, if I remember, there was a study done in the Kaiser Permanente healthcare population.
That's right.
100,000 people who had gotten the shingles vaccine, and they followed them, and they could see that every year the efficacy was declining until.
That's right. I remember that now. Yep.
So this other vaccine is very appealing. And yes, so I had heard about that when I visited Pitt, and we did it on TWiV. And then you said that we should get somebody on, and I emailed him, and he said, well, I'm not really an expert on VZV, and then I stopped there. So
Right.
we should get someone on at some point.
Ann Arvin. Is that right?
Yeah. Ann Arvin
at Stanford.
Yep. Okay, that's a snippet of 1 hour.
I knew that wasn't going to make it as a snippet. It's just too good.
Yeah, it's okay. This episode is brought to you by the Defense Threat Reduction Agency. Imagine an everyday, inexpensive drone that you can buy online, which has been modified by terrorists to spread chemical or biological weapons over a crowded football stadium or a holiday parade. Plague, VX, sarin, weaponized flu. How would we treat the victims? What could we do to counter the effect? How can we prevent a scenario like this from happening? These are some of the topics to be considered in Long Beach, California on November 28th through 30th during the 2017 Chemical and Biological Defense Science and Technology Conference, where you can exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Collaborate with more than 1,500 scientists, subject matter experts, military service members, industry partners, and academic leaders from across the globe who are committed to making the world safer by confronting chem biodefense challenges. In case you're wondering, the Defense Threat Reduction Agency is part of the U.S. Department of Defense, and it's charged with safeguarding our warfighters and our nation. Their department is hosting this department, the Defense Threat Reduction Agency's Chemical and Biological Technologies Department, is hosting this important conference, which brings together the best and brightest from around the world. Please join us to share your important ideas. For more information and to register, you can go to cbdstconference.com. And if you want to keep up to date with latest conference information, you can find that on Facebook. Just search for the page DODDTRA. And that's a little acronym of Department of Defense Threat Reduction Agency, DODDTRA. That's on Facebook. Or on Twitter, CBDST Conference, all one word. The 2017 CBD-S&T Conference, today's innovation, tomorrow's warfighter capabilities. We thank DTRA for their support of TWiV. All right. Now, actually, this next paper may have been a better snippet because it's a little shorter.
It is shorter.
It's okay. We can make it shorter.
We can stretch it out too.
We can do anything.
It's a paper in Scientific Reports, so it's open access. I think the JCI paper, Journal of Clinical Investigation, that we just did is also open access.
Yes, it is.
And this is Scientific Reports, which is Nature's open access initiative. The paper is Dengue virus activates C-gas through the release of mitochondrial DNA. And the authors are, let's see if we have some shared things here. I don't see it.
Doesn't look like it.
First author is Bo Sun, and the last author is Eng Yong Oi.
And most of this is happening in Singapore.
Where it's
one contribution from a Duke University lab.
Of course, there's the Duke Singapore thing, right?
Mm-hmm.
Right.
The shared university or whatever it is, National University, Duke National University. So that's a natural. And Singapore, it's very hot.
Yes.
So again, here's for me another shocking thing. What? C-gas sensing an RNA virus, dengue virus?
Yeah. There seem to have been a few papers coming out about this recently. And so it's sort of, the nice flip side of what we talked about before. Suddenly it looks like there are some RNA viruses being sensed by the DNA sensing system. And
this, there was a related paper to this, which is called Dengue virus NS2B protein targets C-gas for degradation and prevents mitochondrial DNA sensing during infection. That is a Nature paper, and that is from Ana Fernandez-Sesma over at Mount Sinai.
Right. That one's in Nature Microbiology, and unfortunately, it's not open access.
Nature Microbiology. Same idea. And then the other is a paper by John Scoggins.
Mm-hmm.
which I'm a co-author on, actually.
Whoa.
Look at that. Let me make sure. Number 7 is he did a massive screen of different viruses which are inhibited. Yeah, pan-viral specificity of interferon-induced genes reveals new roles for CGAS and innate immunity. So what John Scoggins did, starting as a postdoc here with Charlie Rice, and now at his own lab in UT Southwestern in Dallas, he took about 350 plasmids encoding a different interferon-induced gene product, and then had people send him viruses, and then he screened to see which inhibited different viruses. So we sent him polio virus. That's why our name is on the paper.
That's a heck of a matrix.
Huge. And the thing is, when the paper came out, it had CGAS in the title, which I didn't even see that when all the manuscript versions that we had seen. So it turned out in that paper, I think, Brianne, some positive strand RNA viruses were seem to be inhibited by CGAS signaling, right?
It seems like a fair number of flaviviruses. So there's also, of course, some evidence that retroviruses are sensed by CGAS, but that makes sense because of the DNA that's being made after reverse transcription. These are much more curious.
So RNA-inducing CGAS is weird.
Yeah.
Yeah. So let's have we actually, for the Caleb's out there, summarized what CGAS does?
Well, yeah, we did have an episode, but Brianne, why don't you do that? Why don't you do that for us, Brianne?
So CGAS is a sensor that senses DNA in the cytoplasm from different viruses. It is an enzyme that makes a small molecule called cGAMP, and that induces this DNA sensing pathway that will lead to interferon transcription. And cGAMP production is actually pretty important in some of the figures in this paper.
And
go ahead.
What's the role of STING in all of this?
So STING binds to that small molecule cGAMP.
And then that activates the interferon-stimulated gene transcription.
Yeah, that turns on IRFs, the transcription factor, which will turn on the interferon-stimulated genes.
So I just want people to know that's not Sting the singer.
Right. And cGAMP is cyclic GMP AMP.
Yeah.
Which in my mind is, when I first saw that, I thought, this is a molecule from Mars. This is really great, really a weird molecule. So the really fascinating issue here is that we have a pathway that looks like it's designed just to see DNA, and yet RNA viruses seem to be sensitive to it.
Exactly.
So what's going on?
Exactly.
That's why this paper's cool.
By the way, Rich, do you remember the title of the episode when we first mentioned CGAS on TWiV?
Is it a jumpin' jack flash thing?
It's a CGAS, or just gas, maybe. I don't remember. Yeah, something. That was an Allen one. Or maybe it was you.
I don't think so.
So this, besides this background, this paper begins, these individuals have a attenuated dengue type 2 vaccine strain. It's called PDK53, which is really good at inducing interferon. And the interferon inhibits the virus replication, so it makes small plaques. And they found that the plaques are small even in cells that are messed up for interferon signaling. Like what? It's sensitive to interferon, but if you have cells that can't signal the interferon pathway, they're still small. So what else is going on there? They even say in mice that lack interferon alpha beta receptors, these viruses are still attenuated. You would think that in a mouse lacking interferon system, this virus would grow well, right? But it doesn't. So what's going on here? So let's see. The first series of experiments, they look at the relationship between cell density and virus replication. And if you make different cell cultures with different numbers of cells on the plate, the fewer the cells, the better the virus is able to replicate. Which they
so the implication of that is that if the cells can contact each other and communicate with each other through those contacts, then they are better able to defend themselves against the virus infection.
Correct.
Right.
Whoa.
Yeah, well, something's clearly moving amongst cells, right?
Yeah.
Right.
Right. And they have they finger tight junctions.
Gap
And
junctions,
there's a gap junctions, and there's a apparently an inhibitor of some gap junction protein that they can throw into this and show that if they inhibit those junctions, that the cells are less able to fight off the infection. So that says yes, they're probably communicating something important for fighting the infection through these cell-cell contacts.
Yeah, exactly. And this was really interesting to me because typically when I'm studying a lot of these pathways, we study them in macrophage or monocytes, which are suspension cells. So we don't think a lot about the contacts between cells.
But certainly,
this is why you got to listen to TWiV.
Yeah.
I mean, but if something's being secreted, I guess it has to go. The idea is it's not just secreted.
They're talking about direct. Yeah, they're talking about direct contact.
Yeah. Well, is there any time in a macrophage's life when it's not going to contact another macrophage, but maybe another cell, right?
Right. No, absolutely. But I think that in the macrophage cell lines, we don't have this same kind of setup that you would have in adherent cells.
Right. So they immediately think of CGAMP moving through these junctions, which is a big leap, of course, but...
Yeah, there are two papers before this that had mentioned CGAS moving through gap junctions.
Okay. So they, having had these, they probably did these experiments with that in mind. And then, now the next experiment, they look at CGAMP production in infected cells. And in fact, it goes up when you infect. They're using A549 cells, which can make CGAS. It has functional CGAS, right? It can make CGAMP. And they say they add DADT. Now, didn't we just say that it's not a DADT sensor in the previous paper?
Yes, we did. One thing that I think is really interesting that I noticed right away in this figure is the way that they measured CGAMP is very different than what I'm used to seeing. Pretty typically, people will stimulate their cells and then do mass spec or HPLC to actually look at the CGAS in the cytoplasm.
Right.
Or try to transfer cytoplasm from one cell to another and see if you get protein dimerization changes in IRF by Western. So this idea of using this reporter assay was new to me for measuring CGAMP.
Right. So they make a lysate and throw that onto some reporter cells that have been designed to measure stimulation of the interferon response. They turn blue or something like that.
Right. Exactly. And so while DADT is very frequently, described as not being something that hits CGAP or CGAS, there is something transferable in cytoplasm here.
Yeah.
Okay.
But the important part here is that dengue virus infection also increases the amount of CGAMP production, right?
Yep, exactly.
So poly-DADT, that's weird, but dengue doing it is the key.
I was having the same sort of problem with this as Brianne was having. It's not a direct measure necessarily of CGAMP, right? It'd be nice to see the mass spec or something like that.
Yeah. Right. Yeah. So I've seen these same indirect assays, just not with the reporter. But you do usually see mass spec and or HPLC as well.
All right. Now, in the next set of experiments, they actually add CGAMP to cells and show that if you add it before infection, it can reduce dengue virus yields, viral RNA, and infectivity, but not post-infection. So is that Brianne, a good experiment, adding CGAMP?
I think that it is a good experiment, and I think that I like that they show it in comparison to interferon and show that it is sort of working with the same kinetics as the interferon.
So it looks like CGAM can inhibit dengue virus infection, right?
Exactly, yes.
Yeah, and in fact, they knocked CGAS down with siRNA, and that allows for better virus replication.
Exactly. And so that's sort of weird that, DNA sensors here are impacting RNA virus replication.
Right. So if you buy it so far, and I think it's okay that somehow this DNA sensor is impacting dengue virus, what is going on? Now, the next thing they do is turn to mitochondria. And Brianne, what is the reason for that? I mean, I wouldn't have necessarily thought of that.
So there are a few other studies that have kind of gotten into the idea that mitochondria damage or release of mitochondrial DNA might lead to signaling through CGAS. But there's really been a difficulty because it's hard to make cells just release their mitochondrial DNA. So people tend to use some genetic knockouts of different mitochondrial genes or other types of sort of genetic approaches to try to show that mitochondrial DNA can stimulate these pathways. But thinking about how physiologically we could actually get mitochondrial DNA release is unclear. The group from Mount Sinai had also shown that dengue virus might be releasing mitochondrial DNA.
So here they use microscopy and a dye called mitotracker, which I believe stains mitochondria.
I believe so.
They see a mitochondria aggregating in the cells after infection. They find mitochondrial DNA in the cytoplasm. Now, and it's increasing according to the multiplicity of infection. And I think this is detected by PCR. Is that right?
Yes, it is.
Because there is mitochondrial DNA in there, and it's specific and so forth. So you can do that. And if you look at an uninfected cell, you find much less in there.
Exactly. So here they're kind of able to show that dengue does in fact lead to this mitochondrial DNA release into the cytoplasm.
And these cells also undergo apoptosis, which you might predict from cells that have their mitochondria damaged, right? Because they're releasing activators of apoptosis. So the suggestion is that dengue infection is damaging the mitochondria, mitochondrial DNA is released, and then that's sensed by CGAS. But they never actually close the gap.
No.
They never show that mitochondrial DNA can activate CGAS, right?
Right. And so that's sort of always been the tricky experiment. You can transfect in mitochondrial DNA.
Right.
And people have shown that will turn on CGAS, just like transfecting in a lot of types of DNA will work. Or you can genetically mess with the cells to try to make those cells release mitochondrial DNA. But sort of a question of how you do that experiment has been difficult.
I see.
And I wrote this into the show notes. My reaction to this paper was that I wasn't convinced on this paper alone. A lot of the effects are really small. They're statistically significant, but we're talking about much less than a twofold effect, like 25% difference between two numbers. And when I looked at the immunofluorescence, if you showed me those panels and asked me which one was which or whether they were different, at least from what I could see, I would say no. So my takeaway from this was it's a nice idea. The data are not inconsistent with this, but I came away thinking, I'd like to see some independent confirmation of this. And then the other paper that Vincent dug up came up, and I thought, okay.
Are you okay with that?
Yeah. I mean, you put the two. Actually, I like. My apologies to the authors, but I like, in terms of being convinced, the second paper, I think, had more compelling data for me.
So this is the one where they showed that it's a wild-type dengue, NS2B, can degrade CGAS, and that prevents sensing of mitochondrial DNA. Right, Brianne?
Right, they do. They also talked about this at ASV. And if you look a little bit more closely into the paper, it does get at one issue these authors talk about in the discussion. Because you might expect, well, why doesn't the vaccine strain also inhibit C-gas? They say that it has normal wild type NS2B.
Yeah. So it should inhibit it, right?
Yeah, but the other paper actually talks a lot about how NS3 is actually needed for binding to C-gas. And they don't really mention NS3 here at all.
So there could be changes in NS3 in the vaccine strain, right?
Right, exactly.
Interesting.
So this other paper has stuff like basically pulling down C-gas in the infected cells and showing it has mitochondrial DNA on it in significantly higher levels than otherwise. They have immunofluorescence looking at the mitochondria, where it's absolutely clear that the mitochondria look different, and they've got they can probe by immunofluorescence also mitochondrial DNA co-localizing with C-gas in the cytoplasm of these cells. So I found the data in this really compelling. Though, it's always great to have two completely independent observations of the same thing. And that's more compelling than almost anything you can do. So, in the end, I'm convinced that these RNA viruses can turn on C-gas by doing something to the mitochondria that releases mitochondrial DNA.
Yeah, I think it's actually really important also because it is also starting to tell us ways that we can turn on C-gas and inflammation that maybe are things we didn't expect. In terms of maybe cells that have mitochondrial stress from some other cell biological process are suddenly going to have this, what we thought of as an antiviral pathway, suddenly get turned on. There were actually two papers in this week's issue of Nature talking about how replicative stress might lead to some signaling through C-gas.
Brianne, do you think that this is going to be a general feature of RNA virus infections?
Most of the data that I've seen uses some type of flavivirus. I really haven't seen this with too many other types of viruses. So I don't know.
Okay.
That's a good question.
I guess we'll find out. People are going to look at it. Now, why, what is the function of viruses damaging the mitochondria? Because you would think that if that's going to result in activation of C-gas, that would not be something that would happen. But there must be a need for the virus to do this.
I agree, and I don't have a much of an answer for that. Most of what I've done looking at DNA damage and these pathways has involved HIV or other retroviruses, and you can imagine that integration there can lead to DNA damage. So that's the link that I've always seen. But why it would be advantageous for some of these viruses to release mitochondrial DNA, I don't know. Yeah,
I'm not even thinking that it's necessarily advantageous to them. As a matter of fact, this could be it could be some other antiviral sensing pathway or some damage sensing pathway. Is, after all, you get apoptosis down the road, right? Is saying,
right.
There's trouble here, and one of the things that you do is blow up your mitochondria in order to kill yourself. So
collateral
not necessarily
damage,
something. Yeah, not necessarily something that where the virus is trying to gain an advantage by activating mitochondria blowing up. It could be, in fact, the cell's response, one of the cell's many responses to the virus infection. Get rid of the mitochondria.
Of course, if you get rid of mitochondria,
dump the core.
If you get rid of mitochondria, that kills the cell. You don't need to activate C-gas then.
Yeah, well, do everything, right? This is really bad.
But if you activate C-gas, you're making C-GAMP to signal to neighboring cells, and you're making interferon to signal to neighboring cells.
Yeah, so that's the key. You're going to protect those other neighboring cells before they can get infected
Right, exactly. And so that's why that pre versus post is really important that they did earlier in the paper.
Right.
So that's like a virus-infected cell undergoing apoptosis. It restricts virus replication. The cell dies, but the rest of the organism stays.
Right, exactly.
Same idea. Yeah. Makes sense. Nice. Okay.
This is falling on the hand grenade.
Right.
Good title.
That is a good title. I like it.
I thought these two papers were really neat.
Yeah.
And, as I said, Brianne first mentioned the dengue CGAS paper a couple of weeks ago. And independently, I found that VZV paper, and it turns out they make this beautiful arc. And they're both novel mechanisms of sensing, I think. Well, not so novel because Paul 3's been around for a while. Novel to me, and maybe Rich too.
Well, and I think that they're both novel in the sense that it makes us realize that DNA sensing and RNA sensing aren't so separate after all.
Mm-hmm.
Yeah, and you shouldn't get locked into narrow views of things, right?
Right, exactly.
And I think in this dengue paper, they called it our canonical views of sensing. Be careful of canons, yeah.
Yeah.
Okay. Anything else we missed, Brianne, before we move on here?
I think that those are the highlights.
So you work on CGAS sensing, and you use mostly retroviruses, you said, in your laboratory?
We are working on CGAS and IFI16, and we're looking at retroviruses and then we do a little bit with DNA damage and transfected DNA as well.
And you're at Drew, of course, and you use mainly undergrads to do your research, is that right?
Yes. So my research is myself and right now 5 undergraduates in the lab.
Wow.
How does that work?
It works very well. I am very lucky with strong students right now. So for this semester, I teach on Monday, Wednesday, and Friday, and I'm in the lab on Tuesday and Thursday.
Yeah.
And then the students are in the lab as well as they have time.
How long have you been at Drew?
I'm just starting my 5th year.
Wow. It's cool.
Yeah. I really enjoy it here.
Great.
All right, let's do some email. First one is from Cameron. Hi, all. I'm a grad student at Fresno State and wanted to comment on TWiV 451 with regard to Verily's Debug Fresno, California campaign. Just to remind everyone, I'll take a pause here. Verily is a part of Alphabet, which is the parent company of Google, and they have Verily is the life sciences division. And they are releasing mosquitoes in Fresno. We talked about it a couple of weeks ago, and that's called Debug Fresno, trying to reduce mosquito populations. Jody, one of the project leaders from our mosquito control district, spoke about this project as part of a Fresno State guest lecture series this past spring. Aedes aegypti were only recently introduced to Fresno and seem to maintain a strong foothold in a specific part of our city. While they have so far resisted attempts at traditional eradication, hopes are high that this campaign will help drive down the population enough to make traditional mosquito control more effective. I've been explaining the benefits of this campaign to my friends and family in town, and one of the biggest selling points was that only male mosquitoes, the non-biting ones, are being released by the millions. Here in Fresno, we are unaccustomed to the ferocity of Aedes aegypti and will only need to deal with the increase in mosquito numbers short-term until populations hopefully come under control. It's great to see Fresno's name in the news in a positive light, and I'm excited to share a bit of science outreach with my community around this project. On a quick personal note, I wanted to thank you all for the excellent catalog of podcasts. I got my start with TWiV when I was an undergraduate researcher at UC Davis, studying how biting midges transfer blue tongue virus between dairy cattle. Specifically, TWiV kept me entertained as I counted and sorted midges by the tens of thousands. I expanded into the Twix family of podcasts, which kept me sharp as I took a couple of years off before enrolling in a master's program back in Fresno. I'm currently working on a project to characterize the ticks and tick-borne diseases in the nearby mountains of Fresno. I always look forward to listening to episodes while I'm in the field, especially this week as I'll be evading the 108 degree F heat on the valley floor. Thanks again for all you do. Cheers. I remember once driving through the valley so we were in the mountains east in California, was pretty cool. We drove through Fresno. The thermometer went over 100, and then we hit the coast, and it went back down in the 70s. Wow.
Wow.
It's amazing. Yeah. You must know that, Rich, right?
Sure. Absolutely. Yep. A lot of climates in California.
All right. Can you take the next one, Rich?
Steve writes, "Hi, Vincent et al. Thanks very much for having a look at the Byron Hyde enterovirus ME idea for me." So this was the notion that an enterovirus is a trigger for ME CFS, chronic fatigue syndrome. Sometimes and basically we looked into it and we commented on it and decided that it didn't have much substance. Sometimes it can be quite difficult to judge the merit of these sciencey pieces, even when one is used to having to weed them out. When they come from genuinely well-meaning people, one doesn't feel inclined to dismiss them out of hand as one does those that infest the more obvious woo-woo sites. There was a lot of interesting content in this week's podcast, as usual. It must be great to be a young person in Linda's position. To my mind, there seemed to be a high proportion of talented women in science these days. Maybe I'm just looking in the right places. But I'll just stick to a couple of points. You discussed the need for free access to scientific information again. I don't think that many people stop to imagine what a golden age for scientific progress we have entered with the coming of the World Wide Web. When I was at college, it was usual to have to put in a British library request to be sent a photocopy of a scientific paper by post, that is, first, if we were even aware of its existence. Alternatively, a class of 30 might have to wait their turn to borrow the same copy of an expensive book just to write an essay. Then, if you wanted to check the references, you'd have to put in further postal requests for each one, by pay, of course. No way would anyone be able to check more than a few. Could anyone back in the 70s have imagined that they would one day be able to just click a link and read almost anything ever published anywhere? Yes, 3 exclamation marks is appropriate for this in this monumentally significant case. It is mind-boggling. The World Wide Web will be seen to have enabled a blossoming and acceleration of scientific progress comparable with the Cambrian explosion in the flourishing of biodiversity. Let's hope humanity can live to reap the benefits. We really ought to be celebrating this more than we do. Giving a closer-to-home example, what would it be like for the public to try and break down the biased work of researchers with vested interests if we could not even read the papers? This is the position almost the whole public was in before the internet. For the general public, science was a couple of rows of out-of-date reference books that you weren't even allowed to take out of the library, augmented by a handful of popular magazines and books, if you could afford them. Few people were ever in a position to effectively challenge scientists on their own turf, and so a number of them will have been accustomed to being able to say what they liked with little fear of challenge from outside their peer group. No wonder that some are feeling vexed by the paywalls of their cozy little empire are beginning to crumble at last. This really is the dawn of a second renaissance, and more than just paywalls are going to be swept away. Bad science is going too, and good riddance. Another aspect of the paywall culture that needs to go, just as much as that related to scientific papers, this is the phenomenon of the regionally blocked public information video. I can understand the need to make entertainment pay, but it's crazy to hide stuff that needs to get maximum exposure, especially for health reasons. As a perfect example is in your reader's link to the John Oliver vaccines program. I went to look at this and found many YouTube links, but all leading to the owner does not make this available in your region. Oliver needs to realize that this means that people who search for his brilliant piece of public educational material will be able to access all the videos of cranks exposing it as vaxxer propaganda, but not be able to see his crucially important work at all? Could you perhaps get in touch with HBO and ask them to open it up for the public good? Surely Oliver would want it to reach as many people as possible. In the meantime, I have managed to find a link to a copy of the program on Daily Motion. There are some intrusive adverts, but the whole episode is there, so it is better than nothing. Perhaps you can include the link in your show notes for people outside the U.S., and she gives the link. This is counterproductive paywalling, or just plain blocking of important public information resources, even extends into the work of campaigning organizations. I can't understand for the life of me why patient organizations, for example, frequently make campaigning in videos, then expect the people they are trying to reach to pay for DVDs or actually go out to the cinema for special viewings. Are they crazy or what? This is what their donations are for. Why is it that our culture expects everything we do to make money, even if the real price is that few people ever see the results of important educational work paid for by charitable giving? I see and remark on examples of this crazy waste of material and effort all the time, but the material still stays locked away for all but the few who don't really need to see it in any case. I do hope that the producers of an inconvenient sequel have the sense to make it freely available once the cinema release is over, but I'll bet they won't. Do we really only want to save ourselves from environmental catastrophe if we can make money from it? The ultimate irony, and possibly the most inconvenient truth of all. Sorry, I've gone on a bit, but I think it needed to be said. Best wishes to your brilliant team, from Steve in Luton's, Bedfordshire, England. Still looking cold and wet. Still looking like a cold and wet August is on the way. Standard British summer, in fact. So I like this. I like this last bit. It sums it up. Do we really only want to save ourselves from environmental catastrophe if we can make money from it? The ultimate irony, and possibly the most inconvenient truth of all. Very good.
Well, here on TWiV, we don't try and make money from listeners. We just try and make money from advertisers.
So that we can present more material. Yeah. None of it goes into our pockets.
No, it's just, but I understand that, John Oliver, he needs to make a living and so forth, but maybe looking at, in his case, the vaccine program serves a greater good, and maybe you look at that and make it more broadly released, as Steve is suggesting. Great points.
It was a wonderful show.
Yeah. Brianne, can you take, I think it's the next couple of emails?
Sure.
Short.
Zach writes, "Hi, TWiV. I found this paper and thought you guys might like it. The paper is called Acquisition of Phage Sensitivity by Bacteria Through Exchange of Phage Receptors. From the sunny coast in Australia, it's currently 19 degrees Celsius and sunny. Sincerely, Zach." And it looks like there's a note here that paper was discussed on TWiM instead of TWiV on episode number 143.
Yeah, that's right. I have to save some viruses for TWiM because it is microbiology after all.
So what was this?
So this is a Cell paper where they have phage-resistant bacteria, and they culture them with phage-sensitive bacteria, and the phage-resistant bacteria can be lysed, and that's because the receptors are being transferred to them from the sensitive cells.
Whoa.
Okay. So
is that just a direct sort of membrane exchange?
Yeah, it's not DNA, it's protein. And how it's happening, they're not sure, but it's pretty neat.
Cool.
And I think what happens is the sensitive cells lyse, and their membranes are chopped up, and bits of that somehow get incorporated into the other cells, So it's got neat implications. It's a cool observation.
That is cool.
I mean, we could have done that on TWiV, but, we have to spread the love. Why don't you take the next one as well?
Sure. May writes, "Dear TWiV Genius, I am watching the World Athletics Championships in London. Some outbreaks of norovirus are going on in some athletes' hotel. One gold medal contestant, Botswanian Isaac Makwala, was denied to enter the stadium due to his illness. Apparently, he had an episode of gastroenteritis on Sunday evening. Since then, he got better, and the team doctor said he is well enough to compete. His illness was not confirmed to be caused by norovirus, yet he was assumed to be infected with the virus." And then we have a link for some news about this. "The race ended anticlimax." I guess perhaps they mean anticlimactically. "Since the race, all BBC coverage was switched to his illness. I realize this case is hugely dependent on the IAAF policy for sickness. But what would you do if you were the organizer? Would you let the athlete compete? I would say it should be a case-by-case basis, but I would have tried to diagnose if he had an infection to cause this gastroenteritis. This could have drawn a clean line. Best regards." It looks like someone else wrote in about that same
thing, yeah.
And it's a difficult question.
What do you think, Brianne? What would you do?
I don't know. It says here his illness was not confirmed to be caused by norovirus.
Yeah.
Does that mean that they tested and said he didn't have norovirus, or just that it was unsure?
Yeah.
So what's the problem here anyway? Are they worried about him contaminating a stadium?
Yeah, I guess so.
I mean, how many people in that stadium are recovering from a norovirus infection? Quite a few, I would think.
Probably. And I don't imagine that. I would guess that the biggest risk is to his competitors, and they're probably also in that same hotel and have more risk in the hotel than
exactly
in the race.
So apparently he vomited before the heats of the 200 race.
Oh, wow.
But he was able to compete, and they decided to withdraw him, Now, I guess, so, you can aerosolize the virus when you vomit, right? And you can infect other people. That's been shown in airplanes. So I suppose, I don't know what the lockers are like, if it's a common locker room where everybody's in the same place, right? Or if they have private rooms. I guess not, right?
Yeah, probably all in the same place.
So you can imagine he could vomit in the locker room and infect a lot of the other sprinters, and they would get sick. But, for that race, they'd be okay. But if it's a heat, then there are other races as well.
It would be problematic.
I would say, though, you'd have to diagnose it, right?
Right, exactly.
Because if it's not, maybe he had something to eat that's
right.
causing him to, non-viral, and that's why he's throwing up or something.
Or maybe he was just nervous about the race.
Yeah, could be. So, I don't know. Gastroenteritis Sunday evening. It was not confirmed by Noro. So, I don't know. I don't know if we have a rapid test where you could do this, right? Because if you're just before a heat and you throw up, there's no way you can be.
No, not at all.
So, go race.
Anthony has a link to a New York Times article and then a couple of quotes below. I haven't read the New York Times article, but apparently this indicates to me that it was ultimately confirmed as norovirus.
2 of the cases.
But probably.
I don't know what that means, 2 of the cases. Not his, so he wasn't confirmed.
Right, so there was a norovirus outbreak, but he wasn't confirmed to have norovirus.
Okay.
Let's see. About 30 athletes staying at one of the events hotels in London have fallen ill with virus, and a couple of them were confirmed, to have noro by laboratory testing. It was unclear whether he was one of those with norovirus. And he was quarantined for 48 hours. The quarantine was set to end at 2 p.m., too late for the 200 and the 400. Well, I think it's a tough one, but I think case by case, right? And I think you need to figure out if it's noro or not. And if you don't have a rapid test, develop one, because it's not hard to do.
Right.
I'm sure there is one. Anyway, we have some norovirus people listening. Tell us what you think. Okay, let's do some picks of the week. Brianne, what do you have for us today?
I have a pick that is based on a session that I saw at ASM, which is called the Upgoer 5 Challenge. It is a science communication challenge based, to my understanding, on a book called Thing Explainer. Randall Munroe is the person who writes XKCD, the webcomic, and he has a book called Thing Explainer where he explains different technical and scientific concepts using only the most frequently used 1,000 words in the English language. He talks about the Saturn V rocket, and because there is no word for Saturn V rocket, or because Saturn V rocket are not words that are in the most commonly used 1,000, he refers to it as the Upgoer 5.
Which is more confusing than rocket.
Right, that's true. The book is really fascinating, and there was a session at ASM where people had to give talks about their research using only the most frequently used 1,000 words in the English language. They were really fascinating talks. I thought they were a lot of fun, and we're actually trying to put together a similar event here.
That's cool. How do you say, what do you do for virus?
So a lot of them talked about very tiny things that make you sick.
Yep.
Or very tiny things in your cells. I guess cell is on the list because we use cell phones so often.
I work on a very tiny thing that makes your legs not work.
Exactly.
Or I work on a very tiny thing that can make your baby have a small head.
Perfect.
That would work, right?
Exactly.
Rich, can you do it for your stuff?
Oh, man.
Oh, come on.
I work on a very tiny thing that makes blisters all over your skin and then kills you.
Blister, is that one of them?
I'm not sure about blister.
But pimple would, I bet pimple would be. Anyway, that's cool. Yeah, I remember that from ASM. Did you participate in that thing at the ASM?
I did not participate. I was in the audience, but I really enjoyed it. And I'm looking forward to participating in one here soon.
It's cool. So you're going to do that at Drew, right?
We are. We're working on it as an event.
So that's a good reason to go to scientific meetings to get good ideas, right?
Yeah, absolutely. The students thought it was one of their favorite things to see.
Did you ever go to, what's that ASM education meeting called?
ASMQ. I have not been to ASMQ, but I hope to go sometime soon.
I hear it's good. Get a lot
I've heard too.
Good ideas for teaching. Rich, what do you have?
Okay, I get 2 picks. If you can do it, I can't do.
Of course you can.
All right. The furthest, the first is a PBS movie that was just released to celebrate the 40th year of the launch of Voyager. It's called The Furthest, Voyager in Space. It was on last week, but if I don't know what the circumstances are, whether you have to be a member or not, but you can download this from, you can watch it, you can stream it from public television, and eventually there'll be a CD available, a DVD, and they also said you can get it from iTunes. But anyway, this is a 2-hour special that talks about the Voyager missions, and it's just absolutely fabulous.
It's Voyager, man. It's Voyager.
I know. I was thinking that the whole time through. Okay.
You know what that's about, Brianne?
I don't.
Go ahead.
That was the first episode that was the first Star Trek movie, I think, wasn't it?
They found some.
That was about this spaceship that came into the solar system and was wreaking all kinds of havoc. And it kept saying, I forget quite the plot, but it got named Voyager because of its own thing. But because of its own communications, but they finally figured out that it was some aliens had found Voyager and were trying to go back to its origins with the thing.
So it's Voyager without the O.
Right.
Without the O and the A.
So it's really good. It's one of these typical things where they interview a bunch of people that have been who are associated with involved in the program, who are now all 40 years older than when it was launched in 1977. And they talk about the whole mission, and they don't skimp on the science. And the passion of these people just to me was really gripping. It really communicates what science is in a really compelling way. And I was thinking the whole way through, geez, we spend so much energy and so much money on trivial, stupid social and political stuff when we could be doing this, because this stuff is just wonderful and it's all positive. It's all great.
That V'ger is still chugging out there, right?
V'ger is both of them. There's V'ger 1 and V'ger 2, which I wasn't totally aware of, but they built 2 of these basically as backups. And the mission was to go to 2 of the outer planets. Would that be Jupiter and Saturn? That was the primary mission. And Voyager 1 was leading Voyager 2, even though Voyager 2 was launched first, Voyager 1 overtook it. And as soon as Voyager 1 completed that mission, they let it go. And actually, at the very last minute, they turned it to look at one of the moons of Saturn that wasn't on the agenda, but was right at the right place. Okay. But in order to do that, they knew they were going to have to let it spin off into space, and that would be the end of its journey. So they did that. And then Voyager 2, they sent off further past Neptune and Uranus. And it's going out in space now. As a matter of fact, if you click on this link that I've given for the movie, it's got a little ticker down on the bottom that says how far away they are.
Neat.
So I really, if you can somehow go see this, it's terrific. So my second pick is
they're in the billions, I think.
Oh yeah,
they're like over 10 to 12 billion miles.
And of course, a lot of this is about the vastness of space and the fact that they sent those. They interview the guy who made. These things carry this gold record.
Yeah, right.
That it's like 2 hours, and an hour and a half of it is music, and then the rest of it is they got 100 photographs, and then they have greetings in a bunch of different languages. Include the guy who does the greeting from the Children of Earth is Carl Sagan's son.
That's right.
They interview him. Okay. So there's a lot of, they interviewed the guy who made the record. And this guy says, I guess you could say I'm a record producer, but I've only produced 2 records in my life. There were only 1 copy made. I've only produced 1 record. There were only 2 copies made, and they shot them into space.
That's funny. I've heard that. Yes.
So
cool.
That's very good. So my other is sort of an update, if you like. If you'll recall, I had a pick a while ago called a blog called Vipassana Mama, and this is written by my friend from a meditation group that I attend, who has a special needs child who writes about coping with that and the roles of science and her meditation in that coping process. And she has written a new entry to this blog called Science, which is a nice description of why science is important and why science education is important and why science communication is important, and the synergy between science and Buddhist philosophy. So I thought that was nice. And it also contains a couple of quotes from TWiV co-hosts, believe it or not.
You bet. From that TWiV Science March.
Yeah,
episode,
from the
right?
Science March. Okay.
Yep.
So give it a look. I'm sorry. This is Angela, who's a pediatrician, and I'm sorry I didn't catch this sooner. It came out at the end of June, but I was busy amusing myself, being in Oregon and stuff.
Yeah, it's a nice post. I like it.
Yeah, it's very nice.
It's a great post.
All right, I have for my pick an episode of a podcast called Revisionist History, and this is a podcast done by Malcolm Gladwell. Some of you may know him. He wrote that book. What is it called? The famous one. Outliers. Sorry.
Yeah, we had our students read Outliers.
Outliers. It's a good book. And he's a writer and journalist, and he's got the big fuzzy hair. He looks crazy, but it's kind of interesting. And he has a podcast, which I didn't realize, called Revisionist History. And this episode is called The Basement Tapes. He does his podcast in seasons. This is the last episode of season 2, the 10th episode. So he does seasons of 10 episodes. Can you imagine if we did TWiV like that? 1 season a year. How many episodes? 352. Anyway, this one is called The Basement Tapes. And what it is a cardiologist in Minnesota is looking in the basement of his childhood home for a box of data from an experiment his father did a long time ago. So his father was a researcher, and he spent most of his career working on a dietary study, a clinical study, where he took 2 groups of people in some kind of home where people can't leave, so they can only eat what they're given. And one group were given food prepared with normal oil, animal oils, and butter. And the other group got food prepared with vegetable oil and margarine. Because he was of the view that, and this was a while ago, that if you did a vegetable oil-based diet, you would lower your cholesterol and you would live longer.
Mm-hmm.
So when he did this study, it took him like 15 years because he had to do a lot of follow-up. And if you had vegetable oil food, your cholesterol went down, but you didn't live any longer. And so he didn't know why. And he said, oh, crap, I really believe in margarine, but this study doesn't prove it. So, he ended up retiring. He put the tapes in his basement. And, the reason he put them in his basement is because the university said, we don't need this stuff anymore. All these old data, nobody wants them. Let's throw them out. And he said, no, how do you know no one's going to want them? So then years later, someone is looking at the effect of linoleic acid. Is that the acid? Anyway, this is in vegetable oil, and he thinks it's actually bad for you. And he said, I could look at this study and see. And what happens in the end is he got the son to find the tapes, and he went through the data, and it shows that there's some negative effect of the oil that negates any advantage to living. Okay, living longer. So anyway, it's a neat story. It's science. But, this is the kind of highly edited podcast where you have music and, he's got a script and he mixes it with interviews. And people seem to like this very much. It's pretty short. But I liked it because he's got a neat voice and it's an interesting story. And they have a lot of kind of lessons in it. At the end, he asked the guy, do you think your father would have objected to you getting this data? Would he say, oh, it's going to ruin my career and everything I work on? He said, no, my father was a scientist.
Right.
And he said, this is what you do. You see where the data take you, Right. And anyway, it's really cool. I would highly recommend it because it's not very long. And here's the cool part. One of their sponsors is Blue Apron. And the way he does an ad is so different from the way he does it. He has the thing at the beginning, and then at the end of the show, he's somewhere with a guy, and they say, okay, we've made 2 steaks. One of them is a Blue Apron steak, and another one is we bought at a really expensive place in Lower Manhattan, and we cooked them, and we're going to have Malcolm taste both of them. And he eats one. He says, oh, that's really good. And he eats another, and we go, oh, that's off the charts. That's got to be the Blue Apron one. And the guy says, yep, that's the Blue Apron steak. Right. How is that? How cool is that? Anyway. Check that out. We also have a listener pick from Jenny. So Jenny had the follow-up at the beginning about unrest. Hi, TWiVers. I thought you would like this quiz. What piece of lab equipment are you? All right, so you go to this place and you answer questions, and they tell you what piece of lab equipment you are. And Jenny says, I am apparently an autoclave. All right, so I took this quiz, and I'm a PCR cycler. A qPCR cycler, sensitive but clever. You value precision and expertise at the workplace. You always adhere to the highest standards and operate at 105% efficiency. You're a control freak at work, identifying the tiniest of errors and blowing them out of proportion. You cycle regularly in your free time in order to maintain your consistent curves. That's not true. And I get along with centrifuges, and I can't stand old pipettes. Anyway, it just takes 5 minutes. It's kind of clever.
I came across this quiz on Twitter, and I've also taken it, and I'm also a qPCR cycler.
Yeah, they ask you questions that clearly point you in this direction.
Do.
You should take it, Rich. It would be fun.
I'm working on it right now.
All right, maybe by the end.
I'll get to it by the end of the show.
You should. It's very short. All right, that's TWiV 456. You can find it all over the place. Apple Podcasts, which is the new name for iTunes. Microbe.tv/TWiV. Most people listen on their phones or tablets now, and there are apps that you can use. If you do that, please subscribe. So we get lots of people subscribing, and that really helps us get more advertisement. We'd really appreciate it. If you do like what we do, consider supporting us financially. You can go over to microbe.tv/contribute. We have a Patreon possibility where you would, give $1 or $10,000 a month, whatever you can afford, or you can give money, or you can buy things. Just check that out. We'd be eternally grateful for your help. Brianne Barker is at Drew University. She's also BioProf Barker on Twitter. Thanks, Brianne.
Thanks. This was great.
And good luck with the new semester.
Thank you. Hopefully I will be ready to go by Monday morning.
And you'll be back next Friday, so you can tell us everything that happened.
I will, yes.
Rich Condit is he's an emeritus professor at the University of Florida in Gainesville. He's now residing in Austin, Texas. Thank you, Rich.
Actually, I'm a qPCR cycler.
We're all qPCR. Yeah. Interesting.
You're welcome. This is always a good time. It's great fun. And Brianne, is fun being on with you. This is good. Now, small crew, great fun.
Yeah, small is good.
Absolutely.
It's not the smallest we've had on TWiV. Of course, that was 2. Dixon and I started with 2, but shortly we became 3 with the addition of Alan Dove. I'm not sure that we ever went to 2 after the initial ones, but we might have.
There have been a couple of episodes, I think, where there were just 2. I think I've done a couple with just you over the years.
Yeah, the summer people travel, and that's good, as long as I don't have to do it myself.
It's nice we got enough of a crew so that can happen.
Yes, we have a good crew built up now, so we can always do that. I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the Defense Threat Reduction Agency for their support of TWiV. Also want to thank Ronald Jenkins, who does the introductory music on TWiV. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Okay, good. We're all set. Are you on a wireless or a wired connection, by the way?
I'm on a wired connection.
Good. Okay, it looks like you're in the bathroom because you had paper towel dispenser.
No, my office is a converted lab.
Those
stainless steel thing, Rich?
Yep.
You pulled the paper towels out?
Yeah, God, when I interviewed for my first job at Buffalo, there was a guy who had an office that used to be a bathroom. And so the walls were tiled. This was awful.
That's horrible.
The walls were tiled, and you could actually see the outline on the tiled wall where the urinal used to be.
Oh, that's terrible.
Oh, wow.
Yeah, it's disgusting.
That is just inappropriate.
It was disgusting.
Wow.
And the guy was supremely weird as well.
I don't know. I wouldn't take it because it's bad for your psyche and all that, right?
Yeah, no, I wouldn't take that at all.
Hey, my very first office had an autoclave in it. So,
wow,
you're kidding?
Yeah.
Why? Why the hell did you have an autoclave?
It's a long story. Actually, for the first 5 years or so, I didn't have an office. I had a desk and a lab, and that was fine. That was good.
I guess that could work. We all start out with desks and labs, right?
Yep.
Yes.
I was fine with that.
Some of us didn't even have desks and labs.
Yeah, as a matter of fact, when I was a postdoc, Both postdocs. Well, I guess I had a small desk in my first postdoc. Second postdoc, I didn't have a desk. I had, there was like one desk for 2 or 3 of us. And I said, I don't need a desk. I don't want a desk. The bench is where I am.
That's right. You want to be standing up pipetting.
I want to be standing up pipetting. I can have my notebook there, take notes. That's all I need to do.